Biomechanics is the science of movement of a living body, and so we like to think of it as the bridge between biology and physics. So in biology, we learn about the study of life and living organisms, and then in physics, we learn about the study of matter, motion, energy, and force using a foundation in mathematics. But in biomechanics, we're really interested in how physics and biology can be applied together to understand how we move. And so we can ask questions like, how do our muscles, bones, tendons, and ligaments all work together to produce movement? So what are these forces that are being created, and how does the biology within all of this tissue affect it? One of the really cool applications of biomechanics is studying the form and function of muscles. And muscles produce force, which allows us to create movement and motion, or you can say do all of the things that we want to do. But muscle has this really neat structure. So at a large scale, muscle connects to tendons and bones, and as our muscle contracts, it allows us to create motion. But if we look inside a muscle, it has this hierarchical structure that is made up of small muscle fibers that join together to form muscle fascicles, which then join to form muscle. And we can look at muscles throughout the body to see how differences in the way that they're arranged affect their ability. And if you click on this interactive link, you can look at this 3D simulation that allows you to zoom in and out in muscle to look at all the way from the muscle fiber level down to the protein level, how muscle really works and creates forces. And today you'll hear from a few students in the multi-scale muscle mechanophysiology lab. So we're all graduate students that are really interested in the form, function, biology, and diseases of muscle that occur across multiple scales. And so we can use biomechanics principles to understand changes at the whole body level down to the cellular level. So you'll hear today about 3D mechanical modeling to understand changes at the muscle level, and then experimental techniques to measure changes in the properties of muscle using ultrasound, and then imaging and modeling techniques looking inside muscle at a muscle cross-section. And so I'm going to pass it on to V, who will start talking about 3D mechanical models.
Hi, my name is V. I'm a senior rank student in the M3Lab. At VD that give you a brief introduction into biomechanics and why we study muscle in the lab. I'm going to talk to you a little bit more about one of the techniques that we use using magnetic resonance imaging or MRI data to study muscle. What is cool about using MRI is that we can look into the muscle in both 2D and 3D view. And furthermore, we can apply material property as well as boundary condition to actually simulate the movement of muscle in comparison to real life. So in my study, I look into a little muscle called levator valli palatini. In the animation below, you will see this is a muscle sling that goes through your soft palate. And its main function is to pull the soft palate back to reach the back of the throat. And this would prevent airflow come out of your nose during speech. One of a really interesting application that we can use a 3D model for is actually simulate surgery, so that surgeon would have a better prediction in the surgical outcome to prevent complication for the patient. In our lab, we also apply this technique into study of other muscle in our body, such as the ciliary muscle in the eyes, or the muscle in the lower limbs, such as the gastroc and the soleus. Another way that we can utilize our muscle model is to calculate muscle volume of individual lower limb muscle, and then compare this muscle volume to a database of healthy population, and actually generate a heat map to visualize this muscle comparison on top of the 3D model that we generate. So what you see here is that the more blue the muscle appears to be on the heat map, that means this muscle is bigger in compared to a healthy population. The more orange to reddish the color of the muscle, that means the muscle is actually smaller compared to the average healthy population. We can then go a step further and actually correlate the muscle volume measured to the individual sport performance, such as the broad jump, sprint performance, and vertical jump height. In our lab, we have the privilege to work with the UVA basketball team to actually see what is the correlation between muscle volume and vertical jump high performance in basketball player. So here's a really quick question game that I have for you. Put yourself in the role of an NBA recruiter. Which of the players below would you want to recruit for your team based solely on their vertical jump ability? What you have is the heat map of the muscle volume comparison of each of these players compared to the average healthy population. You can pause the video right now. I'm gonna take a break and be right back with the answer. So I'm back, and if you answer E player 3, you are correct. This player has the higher jump performance, so turn out a higher total muscle volume actually does correlated to a better vertical jump performance. And some of the muscle actually show better correlation to other. Why this correlation is important, you ask? This information is very critical for the strength and conditioning coach to actually have a good plan to improve the athlete performance. It also helps physical therapists to work with the rehab plan in case if the player have an injury. So next, Ali gonna talk to you a little bit more about another technique that we use to study muscle in the lab.
Hi, so you guys just heard from V, and she talked a lot about MRI, which is a common type of medical imaging used in biomechanics research. But we can also use ultrasound to look directly at the human anatomy. And when we think of ultrasound, we often think of using it to look at babies in pregnant mothers. But we can also use it to look at muscles and tendons in the body. An ultrasound is a non-invasive and inexpensive method that allows for dynamic imaging of the anatomy. And so what can we measure? We can measure tendon thicknesses, displacements, areas, lengths, and even get an idea of tissue quality. And from all of these, we can then estimate the mechanical quantities of stress, strain, and force to then learn about the material properties of the tissue. So an example would be tracking the muscle-tendon junction between the Achilles tendon and the gastrocnemius muscles, or your big calf muscles, and across ankle angles and watching that displacement and tracking it. So here's an example of an ultrasound image, and we've labeled the structures of interest. So by measuring this anatomy, we can then use the known relationships in biomechanics to determine other properties of the tissue. So one example would be to estimate disease progression based on the muscle quality and size. So these are two images, one of a healthy subject and one of a subject with Duchenne's muscular dystrophy. And Duchenne is a muscle wasting disease. So these muscles get smaller and they get infiltrated by fat and fibrosis. So when we think of ultrasound, it's based on the principle of reflecting sound waves from the tissue back to the probe. And those sound waves reflect at different speeds depending on the tissue that they hit. And then based on that speed, the ultrasound machine converts that to a grayscale level, and that gives us our pixel color in our image. And so when we look at our healthy muscle, it's a lot darker than our Duchenne's muscle. And that's because muscle should be relatively black. But the Duchenne's muscle has a lot of fat and fibrosis infiltration, which reflects back brighter. And so we can see that this muscle has a lower quality than the healthy muscle. We can also see there's a pretty big size difference in the muscles. The Duchenne's patient has a really small thickness of their muscle compared to our healthy muscle, which is an inference of CSA, but we would image that in a different plane to really get an idea of CSA. We can also learn how to really prevent injury by measuring these displacements and calculating strain. So strain, we know, is the change in length over the total length. And when we think about our example from the last slide with the Achilles tendon, we see it displacing across these ankle angles. We can track that displacement and measure it, and then we can divide that by the total length of the Achilles tendon to get an idea of the strain. And we think about strain, we want that low strain because a high strain would mean that it's prone, that tendon is closer to the amount of force where it's going to tear in that stretch. And lastly, we can estimate the force capabilities of muscle by measuring the fascicle lengths and the pennation angle. So here we have two examples of muscles. One has really short fibers, but a large PCSA, which the PCSA is the physiological cross-sectional area, which takes into account the pennation angle along with that cross-sectional area. And then we have another muscle that has long fibers with that small PCSA. And so we can see these two curves on the right, the muscle fiber force and the length, and then the muscle fiber force and the velocity. And these two curves are really principal in the study of muscle mechanics. And we can see that the length of the fibers and the PCSA affect how much force we're going to be able to produce in that muscle. And so from these relationships, we can calculate and make an estimate of how much force this muscle will be able to produce. So we set up a little example for you guys. We have our two competitors, Hunter and Riddy, and Hunter is a weightlifter and does rugby. And that means he's doing a lot of explosive, quick movements. Whereas Riddy, she's a dancer and does yoga. So she's stretched out and holding these stretched positions. And so we know our muscles and our tendons adapt to what activities we're doing, but there's also differences when we have height, and we can see they're pretty different in terms of height. And we can also see that they are different genders, so there's a difference there as well. And so we're kind of exploring all of these differences when we look at this. So our question is, who do you think is going to have a greater muscle tendon junction? Hunter, who does these explosive, quick movements, or Riddy, who's doing these long, drawn out and holding these stretched positions? So to really look at this, we used ultrasound to track the displacement at different ankle angles. And so we see here that I'm ultrasounding the muscle tendon junction at a variety of angles, and V is moving the foot passively because we don't want any muscle action to interfere with the properties of the tendon that we're trying to measure, because we're just trying to isolate that tendon displacement. And so we see in this video here, we're tracking that displacement across the angles. So I hope you guys were able to think of an answer to our question that we posed. But you can see here the results. The answer is Riddy. Riddy has more displacement, which makes sense because she's holding these stretched out positions. So she's going to have a little bit more displacement because she's forcing her muscles to have that greater displacement. And we see that there's about an 8 millimeter difference when we get to that final dorsiflex position. And when we talk about dorsiflexion, that'd be pointing your toes towards the ceiling. So you're really getting that stretch in your Achilles tendon. But we wanted to really look at how the height affected it. So we normalized these measurements by the height, and we can see that Riddy had an even greater percentage of her height displacement compared to Hunter, meaning that she still has so much more displacement than Hunter, which really comes back to her activities that she's doing and her dancing and her yoga. So when we think about this, some questions for you guys to consider is think about strain. Who do you think has more strain? Riddy has a higher displacement, but she's going to have a shorter tendon. But we kind of normalized it here, so you guys should be able to get an idea. And then when we think about this strain, we're thinking about injury. So who do you think would be more prone to an Achilles tendon injury? So thank you guys so much for listening to me. I hope you guys were inspired and enjoyed our little talk on biomechanics.
Hi, my name is Catherine, and I'm a second-year graduate student at UVA. So Allie and V have already discussed muscle at a larger scale, looking at the whole muscle, but I'm going to go into how we look at the muscle at the cellular level. So moving to a scholar scale provides us with a more detailed look at what's happening within the muscle, and it can help us understand how muscle responds to injury, rehabilitation, or exercise through all the cellular mechanisms that are going on. So one of the techniques we use to study muscle is histology as well as immunohistochemistry. So histology is when we look at a tissue under a microscope, and immunohistochemistry is when we will stain that muscle with a fluorophore that will light up, and I'll go into more detail later. So in this example, we'll look at mouse muscles, specifically the soleus, which is a muscle in your calf. So the first step is to harvest that muscle and cut it into very thin sections that we put on a microscope slide. So then second, we'll prepare the muscle for imaging on the microscope by staining our proteins of interest. So the staining process involves adding a fluorophore that will bind to the proteins we're interested in so that it can be visualized using a specific type of microscope called a confocal. So this is kind of what immunohistochemistry is. So in a confocal microscope, lasers are used to excite the fluorophores that were bound to the proteins of interest. So here we use two different stains. One is laminin, which is shown here in green, which binds to the fiber border. And that just gives us the details of the muscle fiber. So then the second one we used is Prussian orange, which shows the damaged tissue. So all of the damage kind of is highlighted in red here. So then after we've used the confocal microscope to take the images, we will process and analyze the images. So here we overlay them, and then we can see the number of healthy fibers compared with the number of damaged fibers. And that helps us better understand, I guess, the damage that's done in the muscle. So there's plenty of other different things that you can do. Just in this example, we're looking at the damage. So I guess a little bit of an exercise for you guys to try out is, can you tell which muscle is healthy and which one is damaged? Based on, these are using the same stains, the laminin for the fiber border and the Prussian for the damage. So I'll give you a sec to, I guess, or you can pause the video. Okay, hopefully you guessed correctly, but the one on the left here is damaged because you can see where the Prussian stained all the damage red underneath the fibers, whereas the one on the right is a healthy, uninjured muscle. So there's really no damage except for, I guess, maybe some artifacts on the side that just happens from handling the muscle. But hopefully you guessed correctly and learned a bit about histology. Next, Megan's going to talk about how you can kind of use this data to inform computational models that will help us understand and predict and maybe plan for experiments that will help further research.
Hi, my name is Megan, and I'm a first-year PhD student in the M3Lab working on modeling sex differences and muscle regeneration. Skeletal muscle regeneration following injury is a complex dynamic process that involves numerous cell types. Following the initial injury, the muscle degrades and the fibers within the muscle experience necrosis, which is the death of cells within the muscle. Inflammation sets in, which causes an increase in inflammatory cells that work to clear the damage in the muscle. Over the next few weeks, the muscle will regenerate and remodel with the assistance of other helper cells and hormones. For acute injuries, the muscle returns back to its initial state in about 28 days. Our lab has developed an agent-based model of muscle regeneration. An agent-based model assigns behavior roles to agents which organize the actions and interactions of agents within the model. Agents can be anything that alters its behavior in response to input from other agents in the environment. The model is run over time and repeated to obtain numerous outcomes for the system. Our agent-based model represents cells as agents, and the input is the injury to the muscle. The model visualization design was based upon muscle histology images. So when we run the model, you can see that the black areas represent individual muscle fibers, and then once the injury occurs, the damage is represented by yellow areas. Over time, the damage causes the fibers to decrease in size, but the cells eventually move in and help remove the damage. The cells then continue to repair and maintain the skeletal muscle, and then those cells can also secrete other factors that help the muscle regenerate back to its original size. It's also important to consider how different sex hormones can affect muscle regeneration. It's hypothesized that estrogen helps decrease muscle degradation with injury and impacts inflammation differently depending on the levels in the blood and muscle. We can test how these theories impact muscle regeneration by modeling these alterations in the regeneration process and analyzing the outcomes.
So from all the cool studies you've just heard about, we know how to measure changes in muscle properties and build models, and this allows us to answer new questions, such as relationships between muscle structure and function. And so we heard about experimental measures such as ultrasound to measure the structure of muscle, and we also heard about complex 3D models to recreate muscle structures and predict their function. And so with these techniques, we can measure changes in structure and function to determine relationships between the two. We can also think about ways to improve muscle function. So when we compare subjects that have more atrophied or weaker muscles with more hypertrophied or stronger muscles, we can think about parts of their training programs that may relate to these differences. We can also think about how therapies might influence muscle properties. So we saw images of healthy and diseased samples, but we can then look at samples that we apply treatments to see if we can restore them to the healthy condition. And so overall, mechanical modeling of muscle allows us to do a lot of things, and it all comes from a biomechanics basis.